Hello guys, this is the last set of video notes for the DNA unit. So we're just going to start by a little review. This is the whole process of protein synthesis. It includes DNA, which is through the process of transcription, mRNA is made. Through the process of translation, a protein is made. Okay, so that's just your basic flow. This is a little new diagram I downloaded from the internet. So it shows your double-stranded DNA through transcription is made into RNA, and remember that's the mRNA or messenger. It leaves the nucleus, travels to a ribosome where translation occurs, and these little circles are amino acids. And you put a bunch of those together and you get a protein. So that's kind of where we've been. Now I want to do a little bit of a review to refresh your memory. Which one of these? This process here, which would be their number one, this little gray circle, number one, or this process here, number two, or number three, this process here. Which one of these three processes shows DNA? Hopefully you said this one, the double-stranded one, the one that looks like a twisted staircase or twirly ladder. That's your DNA. Between this process here and this one here, which one is transcription? When you turn it into a verb, it becomes transcribe. So is that the middle one or is that the one on the end? Well, transcription, which is printed right here, is when you use DNA to make mRNA. So the DNA is this dark blue. Here it is, untwisted. And then this reddish brown color is a single-stranded mRNA. So this center process is transcription. Translation is when we're translating languages. We're going from mRNA to protein language. So we're going from a nucleic acid to a protein language. This is translation, which is typed right here. It also means protein synthesis. So that makes this big gray blobby thing. That's where proteins are made. So is that big blobby thing rRNA, tRNA, or mRNA? A process that the organelle that makes the protein. If you said that that is the ribosome or rRNA, that would be correct. So this gray blobby thing is the ribosome. Now, this red line here is the same red line over here. So this is your messenger RNA. It's a single-stranded molecule, and the bases are just hanging out. The code is exposed for any virus or bacteria that gets into a cell, can attack and destroy it. So what is this gray thing here? It's number 3. It's got these brown circles on it at one end, and then it's got what's labeled as number 5 at the other. It's a form of RNA. And since it kind of looks like a funny little letter T, this is the transfer RNA. That makes number 4. This is the language of proteins. So what are the building blocks of proteins? Well, these would be amino acids. So these are your building blocks of proteins. They're all bonded together. Now, which has the codon? Is it this mRNA strand here, or is it the tRNA here? So which one has the codon? Codon, remember, is another word for word in nucleic acid language. It is always 3 nucleotides long. Well, the mRNA has the codon. So this is the codon, and then the tRNA has opposite of the codon, and the prefix for opposite is anti, opposite or against. So number 5, this would be the anticodon. So these are the codons, every 3 colors. 1, 2, 3, codon. 1, 2, 3, codon. The anticodon is on the tRNA. And the tRNA brings the correct amino acid to the ribosome. So it is impossible for translation to make a mistake. All three nucleotides on the codon must match all three nucleotides on the anticodon. If all three do not match, it's not going to fit. It's like a puzzle piece. They won't fit together. Like you trying to use the key to your front door on your best friend's front door, because they're not going to work. They're not the same lock. So there can be errors in genes, which gives you something that we're going to be talking about now. So let's look at the very last page of notes, and you're going to see the word mutations. That's the title of the whole page. A mutation is any change in the sequence of nucleotides of a gene. So if you look at the nucleotides here, these little yellow, different colored things that are sticking up in the mRNA, those are the bases. If you change a base from 3 A's in a row, the codon is AAA, you change that to AGA, you now have a mutation. And that could be a problem. So that's what we're going to talk about in this last lesson. Now, some mutations can affect proteins by changing their shape, so they don't work. Okay, so those are the bad ones. An example, some people that are insulin dependent, this person has to inject insulin in himself, and we do have the pens now to where you don't have to actually use a needle like that, but diabetics have to take insulin because there's a problem. And there's a bunch of different causes for diabetes, but one of the causes is there is a mutation in the membrane protein that insulin has to use to attach to a cell to let the cell eat sugar. So all cells have to eat glucose for their mitochondria. And some people, their insulin is not made properly, or the membrane protein the insulin attaches to, there's an error. So that's a mutation or a change in the DNA that makes that problem. Now, some mutations are also silent. They don't change any function at all. And we'll look at that also in this lesson. Now, what are some causes of mutations? Well, we as man, we can do things to ourselves that can cause mutations. One is overexposure to X-rays. This would be like an MRI picture of a human being. But if you get in an accident and you go to the doctor, they think you broke your leg, then you're going to have to get a bunch of X-rays. Well, if you then next week turn around and go to the dentist, and the dentist is like, hey, we need to take X-rays, then your parent should speak up and say, well, hey, wait a minute, last week we just had a bunch of X-rays because we thought he broke his leg or her leg, and the dentist will postpone those X-rays, and they'll schedule it for the next time. You just don't want to get a whole bunch of X-rays. Madame Curie is a woman who discovered X-rays, invented the first X-ray machine. She inadvertently killed herself as well because of that, because she didn't know X-rays were hazardous at the time, like we do now. Also, which everybody knows, overexposure to sun, are called UV rays, can give you skin cancer. So these are different kinds of skin cancers. Basically, any mark or mole or lump on your skin that's not a perfect circle can be a problem. If it's multicolored, notice these have light and dark patches. Light and dark patches. Those can be signs of potential issues. If it's not a perfect circle, if the edges kind of get crusty and or red at the skin around the area gets a little bit red, there's a little bit of redness, then that could be signs of cancer. You would just, the next time you go to the doctor, point to it and say, hey, what's this? And the doctors will, even your primary care doctor, will know, well, you don't have to worry about that. Or they may tell you need to see a dermatologist, which would be a specialist, to kind of make that determination. There's a whole bunch of weird marks, birthmarks included, that are not cancerous. Okay, so this is by far not the only way that they look like, but anything that's just kind of uneven, different colored, gets bigger over time is a big one. Turns crusty or red around the edges. That's just something the next time you go to a doctor, say, hey, what's this? So these are ways we can change our DNA. Let's look at how accidents happen when somewhere in the process that causes disease. So there are two types of mutations that y'all have to know. Now, this first one has another name. Not Pitt County. The state of North Carolina is using the phrase point mutation. Other names are available for this, but your EOC uses the phrase point mutation. And that is when you take out one nucleotide and you swap it for another. For example, these are 3 codons for a gene. And notice each codon is 3 letters. These nitrogen bases or letters are connected to the sugar and the phosphate. So there's 3 nucleotides here, 3 nucleotides here, and 3 here. This line means that there's a mutation along the way. And notice how the second codon has changed. Instead of saying GAG, it says GTG. That gives the disease sickle cell anemia. So a normal hemoglobin, which is inside of our red blood cells, it carries oxygen. This is what it's supposed to look like, little circles. That creates this nice, pretty red blood cell. And here they are floating around in a cartoon bloodstream. Sickle cells. The problem with sickle cells is as the hemoglobin protein in a red blood cell gives away its oxygen, this mutation right here causes a change in shape. So now the round hemoglobin becomes square-like. And the problem is squares can connect together to make long chains, whereas circles can't. You ever try to stack a bunch of circles and they all fall over because there's no angles to hold it. So what happens is in sickle cell, as the cell has less and less oxygen, these long strands form, and notice how this cell is long and skinny now because there's long strands of protein, hemoglobin in there that are the wrong shape. And the problem is when they go from the larger blood vessels to the capillaries, which are your smallest blood vessels, they get stuck, and that causes sickle cell. So that is a mutation of 1 letter. That's all it is that causes sickle cell anemia. Now, the other type of mutation are called frame shift mutations. That is when what we call the reading frame of a gene is destroyed, so the gene doesn't function. And there's 2 kinds. You can add nucleotides to a gene. So instead of having 90 letters long, it becomes all of a sudden 91. Or instead of 90 letters or nucleotides long, you can delete 1, it becomes 89. And now you have a problem. So this picture is here for a reason. Imagine you're in the cafeteria, you're standing in line for lunch. What were to happen if you were standing in this line and the 5 kids in front of you over in this area suddenly changed their mind and walked off? Instead of being back here beyond the picture, you now are going to be where this girl is here because she's got her tray, she's getting ready to get her food. So if those 5 people leave the line, you and everyone behind you moves up by 5 positions. That's what we call a shift in the reading frame. We know a codon is 3 nucleotides long. So when nucleotides leave the chain, then the nucleotides that are coded for, they don't code for that anymore. It changes the order. And I'll give you a picture example here in a second. You can do the same thing in addition. And this may have happened to you already. You're standing in line in the cafeteria and somebody in front of you lets their 2 friends butt in line. So now you're further back in the line than you were before. And so is everybody else that was behind you. So the whole entire line shifts back. That's what we mean by a reading frame. Imagine the people, instead of y'all standing in line for lunch, imagine each of you as a nucleotide, and all those nucleotides get shifted forward if nucleotides are deleted or students leave the line, or you get pushed back because nucleotides or students leave the line. So that's the frame shift mutation, and this is bad. Okay, so example, and this is a real example. So if you're adding nucleotides to a gene, here, what they did, this part here, this is real. This is just an example I picked to make it easier for you to visualize. Imagine a codon says cat, C-A-T, and it says cat, C-A-T, C-A-T, over and over and over and over again. That codes for an amino acid called histidine. So they just abbreviated it with the first three letters, just like we did when we did our protein synthesis race outside. I know it seems like forever ago, right before we got out of school for the virus, we ran outside and y'all had one person ran to a DNA strand and you transcribed it, and another you ran back to your partner and your partner translated it, and then the top teams got candy rewards. Okay, so imagine this is the same amino acid repeated over and over and over again. Now you have added the base. So this is an addition frame shift mutation. You add an A. So instead of this codon reading cat, C-A-T, it now reads ACA. That doesn't code for the same amino acid. Hence, this is now changed. Begins with a T. So instead of beginning with an H, histidine, it begins with a T. Now, the next codon is also messed up. So it says TCA. In fact, every single codon after that addition is all messed up because you've shifted the reading frame. The ribosome reads one codon at a time only. So it must read three letters. When you add a base, the order of those three letters changes. Well, what's a real life example? Well, for a woman's first pregnancy, she's got close to a 50% higher chance of having that baby miscarriage than she will with any other pregnancy. We don't know why, it just is. This is a baby in the womb, and this is the recorded heartbeat. Babies have very fast heartbeats. And this is the baby's head. This is the baby's back. You've got some limbs here. And these white things, that's the first true bone-like structures that are starting to form. So you're starting to get, it's not really bone yet, but it's like cartilage. So it's just starting to form, and that's what you're seeing there. But a lot of times, women usually go in to get their first ultrasound at 3 months because the baby's heart should be beating. And that's what they're looking for. The doctor's listening for that baby heartbeat. While we're talking, I'm going to actually let y'all listen to a baby heartbeat. So you're just going to be able to hear it. This is the actual rate. Okay, their hearts beat faster than ours. But this heartbeat, sometimes doctors don't hear it. And unfortunately, okay, when they don't hear it, that's a problem. And sometimes it's because there is a mutation. We do have a pacemaker in our heart, and there's 2 of them. And sometimes there's a mutation and it gets messed up, and the baby's heart cannot beat for whichever reason. And that the baby's heart will never beat, of course, that baby usually ends in miscarriage, which is Mother Nature's abortion. So it's the first pregnancy only, you have about a 50% higher chance of having a baby with a heartbeat that doesn't function in that first pregnancy. And again, we don't know why, but it is. Okay, so that would be an example. Here's how it works. So this is what's in your notes. You have underneath your addition frame shift, the definition is easy. You're adding nucleotides to a gene. So here are 2 codons, AAT, TGG. Well, after the mutation, the arrow's the mutation, the first codon's fine. but a T has been added. So instead of the second codon reading TGG, it reads TTG. That's going to change the amino acid. And you have a G hanging out here in no man's land. It doesn't have anything to pair with on this example, so it just kind of hangs out. If this is a real example, a gene is not just 2 codons long. A gene is 90 or so, and even more codons long. 90 is kind of short, actually. So what happens is this G is now bumped back 1, and every single nucleotide that comes after this error, all of them are going to be in the wrong order. So it's going to look like this. These red boxes or orange boxes are because they're all wrong. So that's why the gene doesn't function. Now, a deletion frame shift mutation is the opposite. That's when you remove a nucleotide from a gene. So here it is. You have AAT is the first codon, TGG is the second. After the mutation, the A becomes a T. So now AAT becomes ATT. Well, ATT in DNA language happens to mean stop. So this gene will never be translated. It's never going to become a protein because when the ribosome sees the stop codon, it stops, just like we did it in our protein synthesis race. So that means that all the nucleotides after this deletion are all in the wrong spot. Kind of like a bunch of people leaving the lunch line in front of you. Now, here's a real disease. Huntington's disease, we will talk about when we get to genetics. It is a deletion. And it's not just, it's up to 5 genes that are messed up. So it's anywhere from 1 to 5 genes where there's deletion that's caused anywhere from 1 to 5 genes to no longer function. This is a normal person's brain. This is a person with Huntington's disease. Notice the center of the brain, there's a lot of death here. Okay, there's a problem. Now, Huntington's is pretty horrible. You live the first 40 years or so of your life completely normal. The disease lies dormant until one day it wakes up and it causes this. We don't know what activates it. There is no cure. It's a rough, tough disease. And we'll talk about that more later. But it is a deletion, which affects anywhere from 1 to 5 genes. So those are your 2 mutations. Point mutation, which is the term the state of North Carolina uses, and a frame shift mutation, which you're adding or deleting nucleotides, which changes all the gene function. Now, every now and then, a beneficial mutation occurs. Beneficial means a mutation that helps survival. The example, we know where sickle cell comes from. And believe it or not, Africa is not the only place. India, Saudi Arabia also have sickle cell, parts of them. It just so happens that if you are a carrier for sickle cell, you have 1 normal gene and 1 mutated gene. Having 1 normal and 1 mutated gene means you are immune to the disease malaria. Now, if you've been listening to the news, malaria is really big in the news right now because we're learning that potentially the drug that we give people to fight off malaria may actually help people survive corona. We still need testing, okay? So the testing is ongoing. We are doing testing in the United States right now with this drug, but if you've, like, I know malaria, I've heard about that, then that's where. It's in the news right now and it's a big topic. So if you are born 1 normal and 1 mutated gene, you're immune to malaria. The reason is, this is a real red blood cell. Your red blood cell is bumpy, and the organism that causes malaria cannot land on your red blood cells and inject the eggs, which then over time cause disease. They can't land on the red blood cell because if you are immune to malaria, your red blood cells are bumpy, and the critter, which gets injected into you by a mosquito, not found in the United States, guys, can't get malaria in the United States. The organism can't land on the red blood cell to lay its eggs. So you're really, they don't get malaria. Doesn't mean their blood's not contaminated. The critter will live out its life, and when it lives its life, it dies. Okay. However, if you get 2 mutated genes, one from each parent, that does not give you immunity to malaria. And this is another real picture of a sickle cell. It gives you sickle cell too. Currently, and since the 1950s when we started testing, 90% of the Central African, population is immune to malaria. 90% of them. So that means 10% are not immune. Those are the 10% that die. They can die from malaria, or if you have sickle cell, not only can you die from malaria, you can die from sickle cell. And that is mostly because they don't have access to medical care. In this country, few people die from sickle cell because we have access to medical care. Okay? So this is a mutation. We actually know the history. That's pretty rare. We don't know the history of most of them. Okay? Now, let's look specifically at the DNA codons. So this is in your notes. In fact, you actually have this chart in your notes. And next to that chart, you've got this. This is the first one. You're going to look at a codon. You're going to look up in the codon chart AAA and write down the amino acid. Then you're going to look up in the chart AGA and write down the amino acid, which your notes are already done, so it's already there. But look it up to prove it. So over here, AAA. My first letter is an A right here. So that means it's somewhere in this line right here. The second letter in the codon is an A. So that's up here. So that means it is in this box. The third letter is also an A. So am I going to use this A up here? Hopefully you said no. We're going to use this A right here because it is a third letter to an A, which is in this row right here. Those are the only ones we can use. So AAA is lysine, Lysine. AGA. Okay, so we're still with A, but now the second letter is G. So now we are over here. Third codon is an A, and we're not using this A. We got to use this one here. So in this mutation, lysine changes to arginine. And in your notes, does this change the amino acid? Yes, it does. So if this mutation was in a gene, it would change an amino acid, which then may very well make that gene, it doesn't work anymore. Whatever it's supposed to do, it doesn't do it because it's not going to work. Now, the second one you're going to do is the codon GUG. You're going to look that up. It mutates to GUU. So look up these two codons. Yes, it's already written for you, but please verify. So GUG. G is the first letter in the codon, so we're only down at the very bottom. The second letter is a U. So that's this first box here. So guess what? Every single letter, GU whatever, it doesn't matter. It all says valine. So GUG, actually, we don't use this G here. We use this one down here because this G correlates to this first letter being a G. So GUG says valine, excuse me. GUU is the other one. GUU. G, U, and U, third letter being U here, also says valine. So does this change the amino acid used? Nope. So this is what we call a silent mutation. Okay. On average, a human being at least the last data I checked, this is an older fact now, anywhere from like 13 to 75 of these silent mutations in every single human being. So it's a mutation, but as we talked about this codon chart before the break, there's repetition. So all 4 of these say valine. So it doesn't matter in this case what the last letter of the codon is, because they all mean the same thing. Okay? And if we look at this chart, look at this box here. This is the GC box. They all say alanine. Okay? So there's repetition in the chart. So that sometimes is why G mutations are silent. Okay? So just to recap, there's 2 kinds of mutations. What North Carolina calls a point mutation, which simply replaces one nucleotide with another. So AAA becomes ATA. And a frame shift mutation, where you either add or delete a nucleotide which hoses the gene. Gene's not going to function. Okay, so